Hello everyone. I'm really glad that we have the opportunity to gather here again for this year's um, Healthcare AI Forum. Um, just to reintroduce the idea of Healthcare AI Forum, the idea is that we're facing with so many advances in AI, let's say Gen AI, large language models, every week, if not every day, and then it's quickly exceeding anyone's capabilities to assemble all those informations and assimilate and use them. So we wanted to provide a venue that we provide the short and bite chunk of video presentations so that people can take away the intuitions and think about how they can apply those advancements within their own tasks. And over the past year, I had the great pleasure to organize this event with many people that are on this um, webinar here, and including Nam, Meg, and Eva, and Abel. And I've also heard from multiple participants that whether we could extend this event to not just within the greater Chicago area, but also to many other universities. And I've also got interesting collaborators from other universities wanted to contribute to this. So as the chair of the advisory group for this Healthcare AI Forum, after discussion with my co-organizers, we are changing this year's format so that in one of the future sections, we're going to feature each of the participating universities, and they are going to take the charge of presenting and selecting which state-of-the-art developments that they are most interested in and then share with the broader community here. But for this session, we actually have a mixed mode so that we have presenters from Northwestern University. We also have a presenter from Stony Brook University. And so we're gonna try this format for this next year, and hopefully that's going to generate broader interest and stimulate more exciting discussions. Now, without much further ado, let me introduce the three speakers here. So we're going to have Saki talk about how to use large language models to facilitate quality reporting in the health system. And we're also going to have Kaleigh. She's going to talk about how we can use protein-protein interaction network to facilitate tasks like drug repurposing. And last but not least, we're going to have Matthew joining us from Stony Brook University, and he's going to talk about DNABERT, which is the system their group developed. And as the name suggests, it's one of the large language models that can facilitate better understanding of the genetic landscape. So now, without much further ado, let me turn the stage to all our student presenters.
Okay, thank you. I think I'm going first. Let me get my screen shared. All right. So I will be presenting this paper that was published last month in Nature Biotechnology, which, as Dr. Luos kind of mentioned, we're looking at protein-protein interactions on a global level and trying to see how disease mutations in the genome can affect some of those interactions. So why is this kind of important? As we're in an era of big data and trying to integrate all of this multi-omic data, precision medicine has really focused on whole genome and whole exome sequencing. And when we have all of this data from the genome, it's really great. It tells us a lot of information about the genetic mutations happening, but it doesn't necessarily translate to what is happening on the protein level, or specifically what is happening when two proteins are complexing with one another. We also have a lot of data from the transcriptome and the proteome, which can be very useful. But predicting the effects of mutations in any of these areas on the downstream signaling effects is very difficult. We only have experimentally determined models for about 9% of the human proteome, which is really small. There's a lot of experimental challenges with determining crystal or NMR protein structures. They typically require really high-powered detergents, and that's going to disrupt the native protein conformation. So if we don't really know what the native protein conformation is, the experiment isn't very useful. And homology models are pretty limited in this area as well. So that's kind of where this paper comes in. They want to use AI to solve some of these protein-protein complex structures that are unknown, so we can understand the effects on a much larger interactome scale. So they are using AI, but there are still challenges with using AI to predict protein-protein interactions on a very large scale. AlphaFold and AlphaFold multimer, fold doc, all these other AI techniques are very useful, but they're not necessarily scalable to model the entire human interactome. For example, AlphaFold multimer can only generate high-quality models for about 2% of the protein interactome when there's no known homologous structure present. Additionally, each protein is directly interacting with 10 to 15 other proteins, which makes determining the effects of a mutation very difficult. So here, if we have this one protein with a disease mutation in this light red on the right, these are examples of the proteins in the network that could be affected, but we really have no clue which proteins are affected versus others when there's so many connections to a single target protein. So that's kind of what this paper wanted to solve. They wanted to generate the next generation of a protein-protein interaction prediction software. And so they do this through integrating genomic scale data with structural proteomic analyses through the use of 4 deep learning architectures. The first is this structure-structure model, where they have both the structural information for the target protein and the partner protein. The next is a sequence-sequence model, where they don't have any structural information about either interacting proteins. They're just using sequence information and kind of incorporating the technologies of AlphaFold to accurately predict what a structure might be. And then the last 2 models are a combination of structure sequence and sequence structure, where we have structural protein information for one of the interacting pairs of this complex, but not the other. And so what this Pioneer software does is it outputs these interface mutations that are kind of shown in red. And so the interface is the areas of this protein that are essential for binding to another protein. So if we have a mutation in this area that could affect protein binding with the proteins that are supposed to be in complex, that could be very important to know. So after they created their platform, they wanted to validate this against other existing interactome models. So these authors had previously published this Eclair structural framework, which is very similar, trying to predict protein complex interactions. And they see that Pioneer outperforms all of these other softwares in both the structure and sequence-based models, where the structure-based models have slightly better predictions. And so having structural information is still really important to getting these accurate protein complex results. They can do this with the sequence-based models at a much higher degree than the other softwares that exist currently, but the structural information is really important still. So next, they kind of wanted to test their model in a disease context. So they generated this pretty complex protein interaction network that you can see on the right, where they identified 11,000 different interactions among 6,000 proteins, where there's at least one disease-associated mutation within the proteins. So the different colors represent different diseases, and these highlighted circles, the larger the circle, represents a higher degree of disease-associated mutation within those protein interactions. So the authors really focus on cancer for the rest of the paper. And as you can see, this model highly predicts some very important cancer proteins like TP53, HRAS, KRAS, and is really predicting these to be important hubs of mutation sites. And so in the output, we get this little protein figure shown on the left here, where in purple and orange are the two proteins that are complexing, and then shown and highlighted in this green color right here is where the proteins have mutations at the interfaces of binding. So they kind of go to further validate some of this in cancer using the data from the Cancer Genome Atlas. And they want to see, are these mutation rates occurring more in these areas of the protein that are really important for binding, or are they just in other areas of the protein that may not be as important? So they compare mutation rates at the interface versus the non-interface across 33 cancer types and found enrichment for mutations at the protein binding interface in 31 of the 33 cancer types. So just kind of highlighting that their Pioneer software can accurately predict important mutations, specifically in a disease context. And so next, they're kind of looking at survival rates among some of these mutations. And so on the top, again, we have these predicted protein structures with highlighted in green, these areas of mutation important for protein binding. And then on the bottom row, we have some survival curves. And so for pancreatic cancer and soft tissue sarcoma, the mutations shown in this top row are not good for the patients. They result in significantly worse survival probabilities. But in the case of prostate cancer, right in the middle, these mutations are actually beneficial to the patient and result in better survival probabilities. So this software can really predict both important mutations in proteins that we need to target therapeutically to improve patient survival, and also in some cases, beneficial mutations that are occurring in the genome. And the authors do quite a bit of experimental validation for their computational models. I just have this one figure here, but they did do quite a bit of high throughput assays and yeast-2 hybrid screens and showed that the mutation rates that were predicted with Pioneer were similar to what they were seeing in their experimental results. So here's just one example with CCND1. This is a protein that is involved in multiple different cancer types, and it has two interacting proteins, CDK4 and TSC2. CDK4 binds through this little green residue right here, and TSC2 binds at the pink residue. So they performed a co-immunoprecipitation experiment with a Western blot to kind of see if this binding interaction was observed. And so in this pink box here, we can see when we have this mutation at the 162 residue, we lose the binding of TSC2, and the same for CDK4. When we have the 114 mutation in CCND1, we lose that binding. So this is just kind of experimentally validating that these mutations are important, and Pioneer is predicting very important mutations in the protein interfaces that impact the formation of some of these protein complexes. So kind of coming to the conclusions of this work, these authors developed a comprehensive multi-scale interactome framework to accurately predict the interface of protein interacting complexes. And this is really bridging the gap between genomic and structural proteomic analyses. They developed the software into a very easy to use web-based server. I focused on their data in humans just because that's probably the most relevant to all of us here. But they did validate this in 7 other example organisms. I think E. coli had the most data in its interactome, and so they were really able to bring together some of that information. But with the human interactome, we have such limited information that this is really a big step forward here. Even with this technology, the human interactome is far from complete, but it kind of highlights how AI is really going to improve our ability to integrate these large, very complex multi-omic data sets, where we're dealing with a lot of data on a lot of different scales, and combining those scales can be pretty difficult. And mapping these protein-protein interfaces, specifically in the context of disease, can guide our therapeutic advancement. If we know what protein complexes are not forming in the context of disease, we can start changing our therapeutic methods based on that. And so that is about all I have. I would be happy to take any questions that anybody has.
Thank you, Kaleigh, for the great presentation. Very clear. Any questions from the audience? So I can start with one, or I see there is a questions in the QA. And so I think Lorenzo asked to say, I'm looking at 2,000 genomes, 2,000 proteins. A web page isn't scalable. What can we do?
Sorry, could you repeat that?
So
2000.
Lorenzo asked, say, I'm
Oh,
looking at 2,000 genomes, 2,000 proteins. A web page isn't scalable. What can we do?
yes. So they have the web page if you're looking at a much smaller set of interactions, and they have a downloadable software as well. So if you wanted to scale this up, you can. But I don't think you would go through the web server to do so. I think you'd have to do a lot more coding behind that.
Okay, so any other questions from the audience? So in the meantime, I'm seeing a couple of them. Is this an open source framework? One of the,
Yes,
yeah.
yes, it is an open source framework. I believe everything is available from their Nature paper to download.
Okay, that's good. And then I want to ask the question that, so nowadays you can, of course, use protein-protein interactome to better understand the disease mechanism. And now that people heard that the Nobel Prize was awarded to, part of it was awarded to the AlphaFold 3 and design proteins that can target those defects. And so what would you think would happen going forward if we can combine both capabilities?
Yeah, so they do a little bit of combining AlphaFold into their framework, specifically when they don't have any structural information about either interacting protein. They do kind of rely on some of the AlphaFold framework to predict from the sequence what the protein structures will be. But I imagine more of this integration will happen. They again only focused on a small portion of the interactome of what was already published, like in the PDB and what experimental structures we have. So I imagine future work will be headed more towards the structures we know nothing about and trying to get a higher accuracy of a predicted structure from just the protein sequence. AlphaFold is really good at that, but I don't know that it can necessarily it can't scale up in the way that this paper wanted to scale. So if you were looking at a set of two proteins interacting and you wanted to really just focus on those two proteins, the authors kind of said stick with AlphaFold for now. But if you want to look on an interactome context, that's where this software is really useful.
Okay, that's great. So now we are at the time for our next presenter, and thank you, Kaleigh. And our next presenter is Saki.
All right, hi everyone. Today I'll be presenting on this paper published in New England Journal of Medicine AI a few weeks ago. It's titled Large Language Models for More Efficient Reporting of Hospital Quality Measures. And this paper is novel because this is the first paper that they claim to use large language model in the context of hospital quality measures. Slide. So quality reporting is important because it's a necessary thing to have to ensure we're providing good quality of care to our patients. However, it is very expensive and time-consuming. In 2022, U.S. acute care hospital was estimated to cost more than $5 million US, and it required 100,000 person-hours of work. And despite such massive burden, quality measures are often assessed on a small subset of patients, which limits statistical power. And these limitations were manifested in March of 2020 when, during the COVID pandemic, the CMS granted healthcare organization relief from quality reporting so the healthcare delivery system can direct time and resources toward caring for patients. But this also came at the risk of not being able to monitor quality of care. So sepsis is an example of a health condition that has a quality measure. Sepsis is a life-threatening condition that occurs when the body's immune response to an infection triggers widespread inflammation, and this can lead to tissue damage, organ failure, and if untreated, death. So it's a pretty serious health condition. SEP1 is the quality measure for sepsis, and it's short for Severe Sepsis and Septic Shock Management Bundle. And it's a quality measure established by the CMS to improve recognition and treatment of sepsis. And basically it provides guidelines for timely and effective management of sepsis, and it outlines the first 3 or 6 hours of patients' presentations with sepsis. Example would be initial lactate measurement, whether the physician did some blood cultures or if they administered broad spectrum antibiotics or not. So SEP1 will be added to the hospital value-based purchasing program starting in 2026. However, it is met with resistance by doctors because this measure is not very popular. The reason is that there's 63-step complex abstraction processes that requires manual chart review, which no one likes to do. So the problems are apparent. It's burdensome for the physicians, and also there's variability in the answers by the physician just by the complex and nuanced nature of these questions. Here in this table, it's a screenshot of the example of the questions asked in the SEP1 measure. So it involves questions with medical knowledge as well as the temporal nature of the clinical course of the patient as well. So this study was set in University of California, San Diego Health, and this illustrates the burdensome workflow to administer the SEP1 measure. So the abstraction involves was an initial determination by non-clinical analysts from an external vendor, followed by a reviewer from nurses on the quality team, and finally then by a physician review. So the goal of this paper was to use large language models for more efficient reporting of hospital quality measure, and specifically in this case, SEP1. And again, this is the first study to explore capabilities of large language models in the context of hospital quality reporting. The primary outcome of this study was to use SEP1 measure of agreement on category assignment between the LLM system and the human abstractors. The assignment involves pass if they're compliant with the SEP1 measure, fail if it was non-compliant, and out of measure if the patient's clinical case did not meet the inclusion criteria of SEP1. And for this, to assess this outcome, we would need a robust evaluation system, which includes both quantitative and qualitative ways. So in this study, to assess between LLM versus human quantitatively, they looked at Cohen's kappa with two-sided test. They also did a random 10% of cases of which the LLM and human abstractor agreed and evaluated Cohen's kappa. And for the compliance rate, they determined the 95% confidence interval using the Klopper-Pearson exact method. Qualitatively, they looked at disagreements between the LLM versus physicians, and they were adjudicated by physicians. And lastly, quantitative assessments between LLM versus LLM, they ran the same prompts 3 times to evaluate consistency and reproducibility. And Cohen's kappa briefly is a statistical measure that assesses level of agreement between 2 raters. So in moving into the methods, here is the figure 1 system. This shows a system architecture for this hospital quality measure study. The first, the data layer, So they had the structured data retrieved in from the FHIR version R4 format. Because the medication administration information is not available in the FHIR format, they also had to directly derive it from the proprietary EPIC API. Now moving on to the AI layer, they had the 63-step SEP1 process flowchart translated into Python, and they also use LLM and whatnot. I'll get into more detail for this part later. And finally, they had the app layer, where users can technically go in and provide feedback to the AI-generated responses. They didn't really use this app layer for this study, but it would be good for the future to allow for reinforcement learning with human feedback. So this is a screenshot of what this web application will look like on the front end. So all of these questions, the answer will be pre-filled by the AI, and then the physicians can go in to either update the answer or provide feedback so we can train the AI to do better. The study design and cohort, again, this was in University of California, San Diego Health. They used a sample of 100 cases that was reported to CMS from January to May of 2022. And this involved 2 hospitals and 3 annotators. Now diving in deeper to the LLM part of this. So the LLM was used just to get answers from the unstructured notes. The parts that was the structured data, they did through the sepsis consensus toolkit, which they created for this study. So using the unstructured notes and with LLM, with the structured data, they were able to answer the 63-step SEP1 measure efficiently. Now more the details of the LLM, they used SOLAR 10.75B model. And then no additional fine-tuning or prompt tuning was performed. They utilized chain of thought and few-shot prompting strategies with a temperature of 0.1, which is lower than the default to allow for reproducibility. And then they conducted RAG on patients' clinical notes. Here's an example of the prompt template that they used. So it's very concise, clear. And they allowed for chain of thought by specifying thinking step by step. Here's a specific example prompt for a specific question, which asks if there was any explicit documentation about septic shock. They also provide examples. This will be an example of few-shot prompting to give more context to the large language model. Now moving into the results section. This is a table 1 of the study cohort demographics. Median age was 66.5, more males and females. And as for the measure category assignment, the compliance rate was 42.1%. Now going into the results of each of the evaluations that were conducted. For each of the 100 cases and for each question, they determined the agreement level between the LLM versus human. But also at the category assignment level, they found 90% agreement between manual versus LLM with the Cohen kappa of 0.82. Additionally, by running LLM 3 times, they were able to confirm that it generated identical measure category assignments, which shows that LLM is very reproducible. Now moving on to the compliance rate. So system here, we're looking at a confusion matrix, and system generated here means the AI and manual extraction means human. Out of measure, again, means they didn't meet the inclusion criteria of SEP1 measure. And then here, this shows that AI classified 20 of the cases as a failure or non-compliance, and then 19 of the cases as compliant. So that will give us a compliance rate of 48.7, and the confidence interval was calculated with the Copper-Pearson method. This random 10% of the cases of which LLM agreed on, this is actually appears to be redundant because cohen kappa will always be equal when in full agreement. This is just an example calculation I got ChatGPT to do, but if it's in full agreement, the cohen kappa mathematically is always 1. And then now moving on to the qualitative comparison between LLM versus human. So the 10 cases where they didn't agree on, the physician went in to see why that was happening. And they decided that for 4 of the 10 cases, LLM system was actually more accurate, whereas 6 of the cases, the human abstractor was more accurate. So implications of the study, it will allow for timely auditing and feedback of quality of care, as this is a very efficient method and it's less time consuming. It's also less burdensome for the doctors. And it can increase the number of cases assessed, which will increase the robustness of the measure and have more statistical power moving forward. Limitations of the study, sample size is small. They only did 100 cases, but they argue that it is very scalable. And had to access Epic for medication administration information, and this would require modification to support across other EHR vendors. So this suggests that it's not as interoperable as it should be. Another limitation is that they only tested on one large language model. We should be testing on other large language models as well to ensure that generalizable with other models. And they also claim that there's variability in how the physicians categorize these measures. However, they didn't do any double annotation or inter-rater reliability within the doctors, which could be a limitation as well. With that, I would like to take some questions.
Great, Saki. Thank you for the presentation. Very concise and intuitive. Any questions from the group? So there is one from the chat, Lorenzo again. So I missed the reference to the LLM sepsis paper. Can you please post it here? So Saki, maybe you can post it after the QA session.
Yeah.
In the meantime, any other questions from the group? Oh, one question. So from Bimosen, so why not fine-tune the LLM model?
Well, I think fine-tuning can help improve the performance, but in this case, even without fine-tuning, they did the model's performing really well. So that it suggests that in this case, there's no need to fine-tune. And fine-tuning can be computationally expensive. So if it's performing really well as is, I think we don't need it.
And there is a question from Greg. Have you explored replicating study at Northwestern?
Wait, sorry, can you repeat that again?
So question from Greg. Have you explored replicating study at Northwestern?
Now that would be really cool. I'll be very interested in doing that. So yeah.
Great. And another question, so interesting that they use a quantized model. Was any rationale for choosing solar versus others?
Yeah, in the paper, they mentioned they use solar because of apparently it only uses one GPU. And so I think yeah, they probably didn't have extensive resources. And so for this time, they decided to use solar. But they acknowledge that they need to test with other large language models as well.
Great. And then question from Greg again. What type of setup considerations with Epic would you anticipate?
What type of
setup considerations with Epic would you anticipate?
So in this study, they only use Epic to derive medication administration data. So that's the only part that's tied with Epic. But I know there's also recent studies that was able to convert medication administration to FHIR format as well. So that would, if they didn't use it for this study, but if we employed it here, then technically everything can be in FHIR format and we can avoid that problem of using Epic.
Yeah, definitely. That would greatly enhance the predictability and generalizability of the study. And last question. So in resource limited settings without EHR, is it possible to replicate this study?
EHR. Probably not, because this relies on this measure has so many questions as I've suggested, like 63 questions. And so they had to pull a lot of data from the structured data set. And if we don't have access to EHR, it probably means that we won't have answers to all the questions, which would reduce the reliability and accuracy of the measure itself.
Fair enough. Thank you so much, Saki. And our next speaker is Matt.
Hello, everyone. Let me get my slide pulled up real quick. Okay, so I'm just gonna go get started. So hello, everyone. My name is Matt, and so today I'll be talking about the LLM that our lab developed, DNABERT, for modeling DNA and its application and identification of SNVs that disrupt non-coding functional elements in cancer genomes. So I'm just going to start off with, basic biology, the central dogma. So, as we all know, DNA gets transcribed into mRNA, and this mRNA gets translated into amino acids. So there's a mutation in the DNA that gets transcribed and then translated. Maybe there might be a change in the amino acid sequence, and thus changes in its structure and thus into its function, just like in the previous presentation where it changes the protein interactions within cancer. But if we were to look at the genome, this actually might not be the full picture, right? So there's the coding region of the DNA actually makes up about 1.5% of the entire genome. However, the non-coding region makes up about 98.5% of the entire genome. And so, the question, most people think of the non-coding region as, they call it like garbage DNA or trash DNA because, most people think that the coding region is where all the magic happens. But you can say that there is some important things going on in the non-coding regions, and these important things include the regulatory elements, the cis-regulatory elements such as transcription factor binding sites, enhancers, promoters, splice sites, CPG islands, which can methylate, and all of these can change the expression of different genes and thus, the expression of different proteins. And so we would want to look into the non-coding region. Specifically in cancer, so here I have a plot of the mutational density of hundreds of GBM patients. So as you can see, and these are different cis-regulatory elements. So we have the transcriptional start site, the donor sites and acceptor sites. And as you can see, there's a peak in mutational density around each of these cis-regulatory elements relative to the coding region. So a lot of the mutations are actually happening in the non-coding regions. So we would want to maybe model what is going on within those regulatory elements, since it seems that those are the ones that are being the most perturbed, specifically in cancer. So how do we model this DNA regulatory language? So, we were thinking in the lab, what is DNA structure, right? Like I think in terms of modeling things, you have to think of what is a close, I guess, what has a lot of similarity. So we were thinking maybe DNA could be modeled as a language. And so here are two characteristics of language that are sort of similarly analogous. There's an analogous system in DNA, in the regulatory language of DNA. So first there's polysemy. So in this case, you have two sentences: time flies like an arrow and fruit flies like a banana. These are the same two words, however, the meaning is different, right? So similarly in DNA, you have the same gene, but depending on the context that surrounds it, the molecular context, if there's different histone marks, different TFs that are binding to it, different even mutations, then the expression of that gene can change. So that's one thing. And then secondly, there's long semantic dependencies. So for example, there's the sentence like, he is the man who blah, 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 did it. So we know that this did it corresponds to the man who did it. And even though it's like very far off, we, as people who understand the English language, understand that these two are related. And so similarly in the DNA, we have things called enhancers, and these enhancers target genes that are much farther downstream than the enhancer itself. So we would want to model that as well. And so some very smart people in the lab, Jerry and Jihan, went ahead and created DNABERT and DNABERT2 to model DNA regulatory language as an LLM BERT, and so these are basically the papers I'll be pulling the rest of the talk from, and so yeah. So let's take a step back. So what is a BERT? So BERT stands for bidirectional encoder representations from transformers, and so now this scary schematic right here, it might be a little confusing, but I think the main point to focus on is how this model is pre-trained, and then how it is fine-tuned for the downstream tasks that we want it to do. So the way that the pre-training occurs is we basically give the BERT a sequence of DNA. We then tokenize it. So then basically turning these into, you could think of them as words. And then after tokenizing them, then we mask certain portions of the sequence and a certain proportion of the sequence. So it could be like 50% in this case. And then essentially we give it to the model, and then we ask the model, can you fill in the blanks, basically? So by doing this, what the model will do is start to understand, based on the context, on the sequences that are around it, both upstream of it and downstream of it, hence bidirectional, what exactly is the highest probability sequence that fits in this mask. So not only is it learning the highest probability sequence, it also starts to understand what context in which this sequence might fit in here. A way that I like to think of it is, right now I have a 1.5-year-old that I'm trying to teach how to speak. And so basically what I do is, let's say I want him to learn like some sort of object. So, I hold up a banana, I say, this is and then I, tell him to fill in the blank. And so essentially he's learning how to specifically attend to the fact that I'm holding out an object that's yellow and long and is a banana. And so that's the context I'm giving it. And then he will say banana, and that's how he learns what a banana is. But obviously I can't show a banana or a specific cis-regulatory element to a computer. So instead, I just give it the entire sequence of DNA, and then it starts to learn the general structure and general semantics within the human genome. So that's exactly what's shown here. So I essentially give DNABERT, or we give DNABERT the human genome. It looks at the genome. It after doing the masking, it starts to fill in those words, starts to learn grander relationships, starts to learn exactly what to pay attention to. And then we have this pre-trained now DNABERT. And then from this pre-trained DNABERT, we can then fine-tune it to specific tasks, such as predicting promoters, predicting splice sites, so on and so forth. And so this is a great figure from the paper, where essentially before any pre-training, essentially the model just doesn't know what to pay attention to. And all of the attention is just throughout the entire sequence. And then now, after pre-training, sort of gets an understanding of, where the important sites to look at are. And then after fine-tuning, it has very, high attention towards these certain regions. And then so actually, in this case, we were fine-tuning for P53 binding sites. And so if we were to overlap the predicted places with the ChIP-seq peaks for the P53, we see that there's, you know, one-to-one concordance. It is in fact focusing on those regions where P53 binds. And so just further strengthening this point, these high attention regions correspond very strongly to known motifs. So, you know, we fine-tune it to predict CTCF binding sites as well as IRF1 binding sites. And so looking at where the model is paying the most attention to, the sequences that the model is paying most attention to, we can then, you know, plot the frequency of each of the nucleotides in a given sequence. And so we can see that the motif, so this is, you know, the experimentally validated motif. Here is what DNABERT predicts, or, you know, this is the frequency based on DNABERT predictions. And you can see that they're very, I mean, they're basically the same, right? Same thing here with IRF1. Here's the experimentally validated, and here's the DNABERT prediction. You can see that they're basically the same. So through this pre-training fine-tuning system, essentially DNABERT through the pre-training is sort of understanding almost like the underlying biology, right? Understanding these different motifs, understanding the context of DNA that surrounds it, that allows for these motifs to be picking out like the right places where these transcription factor binding sites would bind, or where their cis regulatory elements occur. And so if you were to even overlap this with the transcription factor binding site database JASPAR, you can see there's very good concordance with about 75% match to the motifs that are in JASPAR, with another 500 or 400 that are not. However, you know, these could either be false positives or, you know, maybe motifs that haven't been experimentally validated yet. So that's really, really great to see. So let's, you know, change gears a little bit here and think of this in terms of disease. So how would we be able to use this in terms of disease variants? So what we could do is essentially we could give it a reference sequence, have them score the reference sequence, have DNABERT score the reference sequence, aka give a probability for what the sequence is. So in this case, we're looking at the probability that this is a CTCF binding site in front of the gene XPC. And so after that, we mutate a certain base. So instead of a G, we make it a C, and then we see how that changes the model's prediction. So in this case, you could see that basically it goes from blue to basically light blue or not existent. So this difference basically says that the prediction that is this a CTCF binding site, yes or no, it changes. It changes basically to 0, right? And so this variant is actually already a known risk factor for pancreatic cancer. So this again furthers that, you know, we could probably use this in terms of saying what exactly these mutants correspond to in disease, and tie these mutations, these non-coding mutations, to functional effects. So here's just another example, specifically in GBM. We see that there's a transcription factor binding site here for TERM22. I just wanted to show this just as like to hammer in the point of how we can make these predictions. So essentially, here's a reference sequence for this transcription factor binding site. The model says this has this is 93%. I'm 93% sure this is a TFBS for TERM22. Once I mutate the sequence, however, it is no longer predicted to be a sequence. So in this case, we would say that this transcription factor binding site is disrupted, and there's low chance that the transcription factor will end up binding to this alternative sequence. So we ran about 700 different fine-tuned models for different transcription factor binding sites, as well as the different cis regulatory elements such as acceptors, donors, promoters. And then, you know, we started to gather these candidate variants that have high biological impact in cancer. And so what we did was we found this target at which there's this mutation from an A to a C at this loci, and within this loci is the transcription factor binding site for EGR1. And so due to this mutation, there's actually a disruption of this transcription factor binding site. And in fact, downstream of this within this transcription factor, this transcription factor binds to a location in the gene RSF1, which has already been shown to be correlated to many cancers, overexpression which and correlated with poor survival. So you could think the logic goes, DNABERT predicts, oh, there is a perturbation in EGR1. The transcription factor is no longer binding at that point. RSF1 expression goes down. And so as you can see, the people with the mutation, the blue line, they survive better because higher levels correlated with poor survival, lower levels correlating with higher survival. So that's one of the findings. And so finally, I guess, in terms of further applications of this DNABERT model for prognostication and things of that nature, essentially the question that we want to ask is, given whole genome sequencing, can we utilize the non-coding profile of a patient to create a single score for them, whether that be, if we're using somatic mutations from their tumor for prognosis, if we're using germline maybe as an early diagnosis tool, and then can we also utilize the predictions and the important features that make up this score to form new hypotheses, new targets for these different cancers, or even new biomarkers, because obviously in GBM, one of the most famous biomarkers is IDH1 or NGMT methylation. And so, maybe we could also utilize different non-coding marks to further and better stratify these patients, and apply this to a myriad of different cancers, essentially adding more information to our assays, right? And then finally, in terms of variants of unknown significance, I guess like one of the quote unquote bad things about GWAS is we have these mutations from GWAS that are, highly related to a disease. And unless they're related using like EQTL or SQTL analyses, we don't really know exactly what that effect of that mutation is. But we feel like, but using DNABERT, we can utilize the power of a large language model to essentially give a name, give a function, give a functional impact to these variants of unknown significance. So with that, I think that's my time, and open to some questions.
Thank you, Matthew. A great presentation. Any questions from the group? I didn't see one in the QA session, so I think maybe I can start with one. So I've used the BERT structure as the backbone for building the model. Have you also thought about other structures like GPT? BERT and GPT, there are differences attending to the left and right context versus attending to the left context only. Just want to get your thought on that.
Yeah, so we were definitely looking into different architectures. I think one of the more recent architecture that has come out, especially when it comes to long range dependencies, is Mambas. And so we were looking into maybe comparing DNABERT 2 to that. In terms of GPT versus BERT, for us, the BERT system, since it is looking, I think they both have pros and cons, right? Since GPT is more generative and BERT is more encoding of that context, we think this is more specialized or BERT is more specialized for this type of task where it's sort of learning. Obviously, they're both pre-trained, but it's sort of learning more of that surrounding context within the genome.
That's great. Thank you. Any questions? Any further questions from the group? So I'm seeing one question coming up in the chat. Where does the normal or control data come from when training these models? For example, ESP for genomics patterns. How do you know a specific data set is normal? So especially for genomic patterns.
Oh, that is a great question. So I think in terms of mutational data, right? The reference sequence, is based on basically like a consensus sequence of thousands of individuals. Now, when you're trying to decide like what's normal and what's not, what you could actually do is look at like the different allele frequencies, and then because some of the I guess some of these non-coding mutations that we're actually starting to find, or at least I'm starting to see, is some of these are related to because these can also the consensus sequence is affected by diversity. So depending on how diverse the set is, you would need to include more individuals so that you have a better consensus set and have that normal set of DNA. And so I think, as these new data sets, these larger data sets come out and we get more individuals of diverse backgrounds, diverse genetic backgrounds, obviously, it'll help to tease out, what is, I guess, ground truth. But, as we know from the Human Genome Project, there's really not that much. There should be a very small difference between these different racial groups and things of that nature. But I think that's also another avenue that one of our lab members is looking into, especially when it comes to germline defects and things of that nature, and basically race-specific risk factors and things of that nature. And I'm very excited because a lot of these data sets are now taking into account all of these different types of individuals, specifically like the 100K or the all of us data set. And I think there's like 500,000 genomes on the European side, from a whole diverse set of individuals. So these are very exciting, and we're trying to get our hands on the data basically so that we can start analyzing it and come up with good normals, right? Because obviously right now it's not as diverse as we would want it to, but yeah, hopefully that answered your question.
Sounds great. Thank you so much, Matt. So we are at the top of the hour, and I want to thank the audience and all the panelists again for the great presentation and for your rapt attention. So due to the holiday season coming up, we will resume our forum in the next year, so in the new year. So we will see everyone again in the new year, and then we will also be presenting some of our partnering universities throughout the sessions in the new year. So stay tuned. Thank you, everyone.